Welcome to our exploration of coenzyme Q10 and L-Carnitine, two critical molecules for mitochondrial function. Both CoQ10 and L-Carnitine are essential molecules that perform critical functions within the mitochondria, the powerhouses of our cells. CoQ10, also known as ubiquinone, is a vital component of the electron transport chain in mitochondria. L-carnitine, on the other hand, is crucial for transporting long-chain fatty acids into the mitochondria for energy production. While both molecules are essential for mitochondrial function, they play distinct roles in energy metabolism. CoQ10 serves as an electron carrier in the respiratory chain, provides antioxidant protection, and is critical for ATP production. L-carnitine facilitates fatty acid transport into mitochondria, helps remove toxic acyl groups, and regulates energy substrate utilization. Deficiencies in either CoQ10 or L-carnitine can lead to mitochondrial dysfunction and various pathologies. These deficiencies are associated with several serious conditions, including neurodegenerative disorders, cardiovascular disease, metabolic disorders, and muscle weakness. Understanding the correlated actions of CoQ10 and L-carnitine is crucial for both health maintenance and disease management. Both molecules are critical for optimal mitochondrial health and have synergistic effects on energy production. They're both involved in protection against oxidative stress, and understanding their interaction is key for developing effective therapeutic approaches. CoQ10 plays a critical role in the electron transport chain located in the inner mitochondrial membrane. The electron transport chain consists of four major protein complexes embedded in the inner membrane. Coenzyme Q10, or CoQ10, is a mobile electron carrier that moves within the membrane between these complexes. CoQ10 accepts electrons from complex 1, which receives them from NADH. CoQ10 also accepts electrons from complex 2, which receives them from FADH2. After collecting electrons, CoQ10 carries them to complex 3, which then transfers them to cytochrome C. This simplified diagram shows the electron flow through CoQ10 in the electron transport chain. Electrons from NADH and FADH2 are transferred to CoQ10, which carries them to complex 3. This electron transport process drives proton pumping across the membrane, creating an electrochemical gradient that powers ATP production. CoQ10 is thus a critical mobile electron carrier in the electron transport chain, essential for efficient energy production in our cells. Beyond its role in electron transport, CoQ10 functions as a powerful antioxidant within the mitochondria. CoQ10 has a unique molecular structure that allows it to donate electrons, making it effective at neutralizing free radicals. As an antioxidant, CoQ10 performs several critical functions within the mitochondria. It donates electrons to neutralize free radicals, prevents lipid peroxidation in membranes, protects DNA from oxidative damage, and helps regenerate other antioxidants. Let's examine how CoQ10 chemically neutralizes free radicals. In its reduced form, CoQ10H2 donates electrons and hydrogen to dangerous superoxide radicals, converting them into less harmful hydrogen peroxide. CoQ10 protects multiple components of the mitochondria from oxidative damage. This includes mitochondrial DNA, membrane phospholipids, and critical proteins needed for energy production. By functioning as an antioxidant, CoQ10 helps reduce oxidative stress, which is especially important in mitochondrial diseases where excessive free radical production occurs. It helps balance reactive oxygen species production, reduces oxidative damage, and supports efficient energy production. CoQ10's antioxidant properties make it essential for maintaining mitochondrial health and preventing oxidative damage that can lead to dysfunction and disease. CoQ10 plays a critical role in sulfide metabolism within the mitochondria. Hydrogen sulfide, or H2S, functions as an important signaling molecule in the body, but at high concentrations, it can be toxic. Coenzyme Q10, or CoQ10, is essential for the proper metabolism and detoxification of hydrogen sulfide. The key enzyme in this process is sulfide quinone reductase, or FOR. SCOR is the primary enzyme responsible for hydrogen sulfide detoxification. It requires CoQ10 as an electron acceptor and catalyzes the first step in the sulfide oxidation pathway. In the detoxification process, SCORE catalyzes the oxidation of hydrogen sulfide using CoQ10 as an electron acceptor. During this reaction, electrons from hydrogen sulfide are transferred to CoQ10, and the sulfur is attached to glutathione, forming glutathione persulfide, or GSSH. Let's examine the complete sulfide oxidation pathway in the mitochondria. The process begins with hydrogen sulfide, which is oxidized by SCORE, a CoQ10-dependent enzyme, to form glutathione persulfide, or GSSH. Next, the enzyme E1 converts Ju-sulfite, or SO3 2-. Finally, sulfite oxidase, or SUOX, oxidizes sulfite to sulfate, SO4 2-, which can be excreted or used in various metabolic processes. CoQ10 influences several enzymes involved in sulfide metabolism. Most directly, SCORE requires CoQ10 as an electron acceptor. Without sufficient CoQ10, SCORE activity decreases, compromising hydrogen sulfide detoxification. CoQ10 also influences the expression and activity of CBS, or cystathionine beta synthase, which is involved in hydrogen sulfide production. Similarly, CSE, or cystathionine gamma lyase, another enzyme involved in hydrogen sulfide production, is influenced by CoQ10 levels. The relationship between CoQ10 and sulfide metabolism has important clinical implications. CoQ10 deficiency impairs hydrogen sulfide detoxification, which may contribute to mitochondrial dysfunction. This connection has been linked to various neurodegenerative diseases, making it a potential therapeutic target for conditions involving elevated hydrogen sulfide or impaired sulfide metabolism. L-carnitine plays a crucial role in energy metabolism by facilitating the transport of long-chain fatty acids into the mitochondria. Let's examine how this transport works by looking at the structure of a mitochondrion, which has an outer and inner membrane, creating an intermembrane space and a central matrix. Long-chain fatty acids cannot cross the inner mitochondrial membrane directly. They need a special transport system called the carnitine shuttle. This shuttle system involves three key proteins: carnitine palmitoyltransferase 1, or CPT1, located on the outer mitochondrial membrane, carnitine acylcarnitine translocase, which spans the inner membrane, and carnitine palmitoyltransferase 2, or CPT2, located on the matrix side of the inner membrane. Let's follow the journey of a fatty acid through this system step by step. First, CPTY on the outer membrane converts fatty acyl-CoA to acylcarnitine by transferring the fatty acid chain from CoA to L-carnitine. Second, CAX-T transports the acylcarnitine molecule across the inner mitochondrial membrane into the mitochondrial matrix. Third, CPT2 on the inner membrane converts acylcarnitine back to fatty acyl-CoA, releasing L-carnitine in the process. Finally, the fatty acyl-CoA enters the beta oxidation pathway in the mitochondrial matrix, where it's broken down to produce energy. The carnitine shuttle system is particularly important during fasting or prolonged exercise, when carbohydrate reserves are depleted and fatty acids become the primary fuel source. This system is essential for organs with high energy demands, like the heart and skeletal muscles. Deficiency in L-carnitine or any of the shuttle proteins can impair fat metabolism and energy production, leading to various metabolic disorders. L-carnitine plays a crucial role in removing acyl-CoA groups from the mitochondria. Inside the mitochondria, metabolic processes produce acyl-CoA groups, which must be carefully regulated. Excess acyl-CoA groups accumulate as byproducts of metabolism. If these build up, they can inhibit critical mitochondrial enzymes. L-carnitine enters the mitochondria and binds to these acyl-CoA groups, forming compounds known as acylcarnitines. When L-carnitine binds to an acyl-CoA group, it releases free CoA back into the mitochondrial matrix. This free CoA is essential for many metabolic reactions. The newly formed acylcarnitines are then transported out of the mitochondria, effectively removing the potentially harmful acyl groups. This removal process provides several critical benefits. It prevents enzyme inhibition, maintains the optimal ratio of free CoA to acyl-CoA, and ensures continuous metabolic flow within the mitochondria. In summary, L-carnitine's ability to remove acyl-CoA groups represents a crucial detoxifying function that is essential for maintaining mitochondrial health and proper cellular energy production. Mitochondrial energy production relies on multiple interconnected pathways. CoQ10 and L-carnitine play distinct but complementary roles in mitochondrial energy metabolism. In the electron transport chain, CoQ10 serves as a crucial electron carrier between complexes. Electrons flow through the ETC, generating a proton gradient that drives ATP synthesis. Meanwhile, L-carnitine facilitates the transport of fatty acids across the mitochondrial membranes. Without L-carnitine, long-chain fatty acids cannot enter the mitochondria for breakdown. Beta-oxidation of fatty acids generates acetyl-CoA, which enters the Krebs cycle along with carbohydrate-derived acetyl-CoA. The Krebs cycle produces electron carriers like NADH, which donate electrons to the electron transport chain where CoQ10 operates. CoQ10 and L-carnitine serve complementary roles in mitochondrial energy metabolism. Deficiencies in either molecule can seriously impact mitochondrial energy production. When both molecules are deficient, the impact on cellular energy production is magnified, potentially leading to mitochondrial dysfunction and disease. In summary, CoQ10 and L-carnitine work in complementary pathways that are essential for mitochondrial energy metabolism. Deficiencies in CoQ10 and L-carnitine can significantly impact mitochondrial function, leading to a range of clinical manifestations. These deficiencies can be classified as either primary, resulting from genetic mutations, or secondary, caused by external factors like medications, aging, or other medical conditions. These deficiencies primarily affect tissues with high energy demands. The heart can develop cardiomyopathy and arrhythmias. Skeletal muscles may experience weakness and exercise intolerance. The brain can manifest seizures, ataxia, and cognitive decline. At the molecular level, CoQ10 deficiency disrupts electron transport in the respiratory chain, while L-carnitine deficiency impairs fatty acid transport into mitochondria. Both lead to reduced ATP production and increased oxidative stress. Diagnosing these deficiencies involves several steps. Clinical evaluation is followed by biochemical analysis, genetic testing, muscle biopsy, and advanced imaging techniques. Blood testing provides key diagnostic information. Normal CoQ10 levels range from 0.5 to 1.5 micrograms per milliliter, with deficiency indicated below 0.5. For L-carnitine, normal values range from 30 to 60 micromoles per liter, with deficiency below 20. Understanding the critical roles of CoQ10 and L-carnitine has led to important therapeutic applications in clinical medicine. These compounds are primarily used to treat mitochondrial diseases, but also show promise for secondary mitochondrial dysfunction, cardiac conditions, and neurodegenerative disorders. Supplementation with CoQ10 and L-carnitine offers multiple benefits for patients with mitochondrial dysfunction. CoQ10 supplementation enhances electron transport, reduces oxidative damage, improves ATP production, and stabilizes mitochondrial membranes. L-carnitine improves fatty acid transport, enhances energy production, reduces toxic metabolite accumulation, and protects cell membranes. Proper dosing of these supplements is critical for therapeutic effectiveness. Standard dosing for both CoQ10 and L-carnitine ranges from 30 to 50 milligrams per kilogram per day. For adults, CoQ10 is typically given at 100 to 600 milligrams daily in divided doses, while L-carnitine is administered at 1 to 3 grams daily, also in divided doses. Pediatric dosing is weight-adjusted, and newer formulations with higher bioavailability may allow for lower doses while maintaining effectiveness. Monitoring during treatment is essential to ensure optimal dosing and therapeutic response. Blood levels of both CoQ10 and L-carnitine should be regularly assessed, along with liver and kidney function. Clinical symptoms, mitochondrial function biomarkers, and functional measures like exercise tolerance should be monitored to assess treatment effectiveness. Current research is exploring several promising directions to enhance mitochondrial therapies. These include novel delivery systems to improve bioavailability, combination therapies that leverage synergistic effects, and development of synthetic analogs with enhanced properties. Additional research focuses on genetic therapies targeting mitochondrial DNA and precision medicine approaches to tailor treatments to individual patient needs. In conclusion, CoQ10 and L-carnitine supplementation represents both established therapies for current mitochondrial disease treatment and a foundation for future therapeutic innovations.